Okay, I believe this is part 9. Moving on to slide 12. Probably not 49. I'll change that for the upload. But bacteriophages. That means going back a little bit in the book. Bacteriophages start on page 338. First of all, we'll look at what is a bacteriophage, and then we'll look at 2 kinds of bacteriophage. The book has 3 kinds. It has lytic phages, it has temperate phages, and it has filamentous phages. We will just look at the lytic phages and the temperate phages. So number 1 is the lytic phage. We'll use this phage called T4 as a model for this bacteriophage. So what is a bacteriophage? A bacteriophage is a bacteria virus. Very similar to the viruses we looked that affect animal cells, inert particles outside of cells, just a bunch of chemicals, active inside cells, either, they take over the host cell machines. They then direct those machines to build replicas of themselves. They steal the cell's nucleotides, they steal the cell's amino acids. All the resources are stolen from the cell. And then the bacteriophage gets out of the infected cell and moves on to new cells. So some important terms regarding lytic phages. Lytic, from the meaning of lysis, lysis meaning to break. You can see here the general scheme of a lytic phage infection. The virus attaches, different slightly to our animal virus, is that the viral particle doesn't get inside the cell. Instead, only the viral genome penetrates the cell. So we have genome entry. We have the co-opting of the cell's machines to build viral bits and pieces, proteins, and replicate the genome. We have the assembly of those pieces together into new viral particles. And in the case of lytic phages, and phages in general, they typically only have the one way of escaping the cell, at least the kind of phages that we're going to look at. They cause the cell to burst, and the phage particles are released into the environment. So a lytic phage, also known as a virulent phage, exits the host when the cell is lysed. The cell is broken open. It's also known as a productive infection because new viral particles are being produced. Our T4 phage is a double-stranded DNA phage that we use as a model. The entire process, beginning from attachment and ending in release, is as short as 30 minutes. And 5 steps there: attachment, genome entry, synthesis, assembly, and release. And I've got a video to show you on that.
The first step in the replication of virulent phage, such as T4, is attachment. Protein fibers at the end of the phage tail attach to specific receptors on the bacterial cell wall. In the penetration stage, the enzyme lysozyme located in the tip of the phage tail degrades a small portion of the cell wall. The DNA in the head of the phage is injected into the cell, and the phage coat remains on the outside. In the transcription stage, The phage DNA is transcribed into messenger RNA, which is then translated into phage proteins. Next, the phage DNA is replicated to make copies for new phage particles. The phage DNA is also the template for additional messenger RNA synthesis. During this stage, many copies of phage DNA are made, and the various phage proteins are synthesized. During the assembly or maturation phase, the phage DNA becomes packaged in the phage head protein, and the various components of the phage structure come together to form the intact or mature phage. Finally, a phage-induced lysozyme is synthesized. This enzyme digests the host cell wall, resulting in lysis of the host cell and release of the phage.
Okay, and then the phage floats off, attaches to new bacteria, infects them, and so on and so forth. So attachment in bacteriophages, pretty similar to attachment in animal viruses. Protein elements on the virus match shape for shape receptor elements on the cell itself. You can see here there's a wide range of things that a virus, a phage, so bacteriophages, you'll often hear them just referred to as phages. A phage might stick to a glycolipid, it might stick to a variety of proteins, it might even stick to the proteins that make up the flagella. In the case of our T4 virus, it attaches to receptors on the bacterial cell wall. Next comes genome entry. This is a little bit different to what we looked at in animal cells. In animal cells, typically the entire nuclear capsid is uptaken in some way by the cell. And then following that penetration event, that's when the viral genome is uncoated. In this case, our bacteriophages have this kind of injection mechanism. We can think of it as an analogy to a hypodermic needle, although in reality it's not quite the same mechanism. But our virus lands on the cell, it attaches through its in this case tail fibers, sticking in receptors, and then it penetrates into the cell interior. It has this kind of this injection mechanism. It goes through the outer membrane, if there is one. It goes through the cell wall. It goes through down into and past the cytoplasmic membrane, and it injects the viral genome directly into the cytoplasm of the bacteria. In order to get through the cell wall, our bacteriophage also produces an enzyme that degrades cell walls of bacteria, is called lysozyme. On to slide 16. We have the viral genome inside the bacteria, and now we have the production of the viral proteins. And in many bacteriophages, there is a sequence of events. There are what are called early proteins and then late proteins. So when the bacteriophage first gets into the bacteria, the first thing it wants to do is to knock out the bacteria's own mechanism for expressing genes. A few reasons why it wants to do that. Number one is that bacteria do have a kind of immune system against viruses. If you've ever heard of CRISPR in relation to gene editing, CRISPR is a bacterial process that allows bacteria to remember past viral infections and then to more easily recognize those infections. So the viral genome gets into the bacteria. There are protein enzymes in the bacteria that can look at the genome. If they recognize it as an invading viral genome, then they can snip it up and prevent infection. So there's this kind of arms race between the virus and the bacteria. The virus wants to get in, make its early proteins, and destroy the host DNA. By destroying the host DNA, it means that the bacteria itself has no mechanism to respond. Or at the very least, it wants to prevent the host from making its own enzymes and its own proteins. So the virus genome doesn't want to be snipped up. Also, the virus genome doesn't want the bacteria making any of its own genes or its own proteins, because it wants all the machines turned to the virus's own benefit. So soon after infection, no host genes are expressed. All of the machines are turned towards making the viral pieces. Then the late proteins are built. These are the structural proteins, the capsid and the tail. At the same time, the viral genome is orchestrating the duplication of its own genome, so its genome can be packaged up and new virus particles can be produced. So once we have all the bits and pieces, we have the phage head and the tails and the spikes. Not spikes in this case. Spikes are more specifically related to animal cells. We have tail fibers that have pretty much the same job. But once all those pieces have been made, they are put together, they are assembled inside the bacteria. So assembly of the pieces. This spike here relates more to the physical point at the tip of the that injection mechanism. And then we have release. Now in bacteriophages there are no enveloped examples. So the virus particle cannot interact in any way with the bacterial membranes. So the only way that the bacteria has to get out is to destroy the cell. Again, the viruses will use their lysozyme. Lysozyme is another protein enzyme that's produced late in the infection, along with the late proteins that make up the structure. Lysozyme then degrades large holes in the wall. Now we had lysozyme degrading small holes. Those small holes weren't enough to damage the cell wall to any great extent and cause it to burst. But now when our virus particles are trying to get out, they do want to burst the cell. The bacterial cell wall is there to maintain the pressure inside the cell. The pressure inside a bacterial cell is about the same as that inside a bike tire. So what happens when you stick a knife into your bike tire? It bursts and all the air is let out. When you poke a hole in your bacterial cell wall, it bursts and all the contents of the cell are released. And along with its cell contents now is a large number of viral particles. So the burst size should be pretty self-explanatory. With T4, it's around 200. That means one virus gets in, 200 viruses burst out. So, the infection mechanism of a lytic phage. Be able to identify and know what happens in each of these steps: attachment, genome entry, synthesis, assembly, and release. Know these terms: productive infection, lytic infection. Let's also add on there that a lytic infection is also known as a virulent phage. Let's also know what lysis itself means, what a burst size is, and know which kinds of proteins will be early, which kinds of proteins will be late, and what those early proteins are trying to do. They are trying to prevent the bacteria from responding, first off, and then they are making sure that all of the machines in the bacteria are turned to viral production, not bacteria protein production.